Hey team, Dr. Jack Gordy here, and today I'm going to be taking you through sensing pathogens. How does our innate immune cells detect those pathogens that are now floating around? And we'll also do DAMPs as well, so that's damage-associated molecular patterns. Essentially, how does the immune system sense perturbations in the extracellular environment in order to induce this inflammatory response here? And here we have a receptor, and this is kind of how it all works basically. We have a receptor here, and this receptor is called RIG, and it can detect viral genomes, so the genome of a virus inside the cell. So this is actually a cytosolic receptor, and here we can see a macrophage that's been infected with a virus, and so its RIG receptors will be detecting the genetic material of that virus and inducing an inflammatory response. So it's just some beautiful images here, but let me just pause and jump into how does the immune system detect those perturbations in the extracellular environment. So this is the inflammation sequence: sensing, signaling, response, resolution, SSRR, nice and easy. And we're going to be covering sensing in this video here. So how do we detect those perturbations in the extracellular environment? Now, a key thing is the pathogen-associated molecular patterns, or PAMPs, right? And this is essentially that the human biology is different to bacterial, fungal, and viral biology, right? They have different molecular structures than we do. We are evolutionarily so distant that we have different molecules that essentially make up our biology. And so that allows us to evolve detectors that can detect these pathogen-associated molecular patterns. So let me give you some examples. So gram-negative bacteria are coated in a lipopolysaccharide, right? So lipo means fat and polysaccharide means lots of sugars. And essentially it is a chain of sugars that is attached to a fatty protein that can then embed into the membrane, right? So the outer surface of a gram-negative bacteria is coated in lipopolysaccharide. Now this is one of the most famous PAMPs, pathogen-associated molecular patterns. The human body doesn't contain these exact lipopolysaccharides, and so it's something unique that could be an alarm signal to our immune system. Now, this is something I really expect you to memorize, because if you read any paper about the immune response, it's likely that they're using lipopolysaccharide to induce inflammation, because it's such a famous and easy way to activate the immune system. It's using lipopolysaccharide over here. Now, yeast and fungus have a glucan coating. That's chains of glucose all joined together in a rigid coating. And those glucans are unique to fungus, different to our biology. So again, it's another opportunity for our immune system to distinguish between our cells and a pathogen cells. Now, viruses have unique genomes. So viruses insert their genome into our cells and essentially hijack our machinery. And essentially our machinery starts to read their genome to duplicate it and create all the proteins that go into a virus. But what's interesting is their genome, and this is an interesting evolutionary battle, has a higher proportion of GUs and GCs in their genome than we do. And so it's an interesting question. Have we evolved to avoid putting GUs and GCs in our genome to allow us to detect the difference between our genome and their genome? So they have a much higher proportion of GU and GC in their genome, and so we have developed the ability to detect those because that's unique to their biology over our biology. So if we look at some examples here, this is actually from SARS-CoV-2. This is from COVID-19. And you have a look at this fragment of their DNA, and it's just GU, 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 GU. It's so GU rich, it allows us to detect that using receptors. And so this is where an important point comes in. So we call these pattern recognition receptors, PRR, pattern recognition receptors. Now, there are loads of them. I could go over millions of them. Basically, for the last 6 years of my life, I've researched one of these, and there are hundreds upon hundreds of them. But I want to take you just through one group of families. Now, they're called TLR, and this stands for toll-like receptor, but the acronym doesn't really provide any information. So I think it's just important to learn TLR. What it stands for, it doesn't really matter. Same with this guy down here, but I'll take you through it. So there are a family of receptors called TLRs, and they have each evolved the ability to detect a different PAMP. So they are pattern recognition receptors that can detect pathogen-associated molecular patterns. And I know I'm hitting you with a lot of jargon here, but this jargon is kind of essential to the field of immunology, so we cannot avoid it. So there's some examples here. So TLR 1, 2, and 5 can detect lipoproteins and glucans. So that's fungus, for example. TLR 4, this is a super important one, probably the most important. PRR, pattern recognition receptor that immune cells express, is TLR 4, and that detects lipopolysaccharide, which is the most famous PAMP, and it's the most widely utilized in research, the lipopolysaccharide. So that's super famous and it's definitely worth learning. So TLR 4 detects lipopolysaccharide. TLR 6 detects flagellants. So flagella are the little spiral things on the tails of bacteria, and so because the human body doesn't have flagella, it's a great thing to detect. So TLR 6 can detect that molecular pattern. Now TLR 3, 7, and 8, and 9 can detect those weird genomes of viruses, right? So those GU and GC rich genomes of viruses, the either RNA or DNA genomes, and they actually, because the genomes of the virus aren't out in the extracellular space, these guys actually sit inside of a phagosome. So the idea is that a macrophage would eat some virus, then digest it, and then the genome would leak out of the virus because it's being digested inside the phagolysosome of the macrophage. So the macrophage has eaten some viruses to form a phagosome. A lysosome has fused with that phagosome and has digested the virus. The genome then leaks out, and we can detect that genome now on these receptors. So you can see why these receptors aren't facing outwards, because the genome of viruses isn't floating around the extracellular space. We first need to crack open the virus before we can look at it. Now, all of the TLRs, they have subtle differences in their secondary signaling pathways, but they all pretty much activate this guy, NFkB. Now, what that stands for is kind of irrelevant. Everyone just calls it NFkB. Now, this is one of those pieces of jargon you just have to learn, because the NFkB is at the heart of so much of the inflammatory and immune response that you just have to learn it, right? Now, it's a transcription factor. So once these receptors are activated, NFkB moves into the nucleus and turns on a whole bunch of genes. Now, there are hundreds and hundreds of genes that NFkB can turn on, and it does depend a little bit on a thing called epigenetics. So on what genes are turned on, but mostly it turns on inflammatory signaling, an inflammatory signaling cascade. So the NFkB translocates into the nucleus, binds to the DNA, brings in RNA polymerase, and turns on all these genes. So we end up with expression changes, and one example of that is cytokines, which is a protein signaling molecule critical to the inflammatory response. So here you can see how we can we have a bunch of receptors designed to detect unique molecules that pathogens express, so pathogen-associated molecular patterns, and these are called pattern recognition receptors, and we have a huge family of those. Now, I know I've given you just a huge amount of jargon, but there's just a few key things I really want you to remember, and that is TLR4, LPS, NFkB. If you learn that pathway there, that pathway is very similar in all the other TLRs and many of the other pattern recognition receptors. So if you learn that as an example of a pathway that leads to inflammation and immune cell activation, that example is relevant to all the other examples. It's often just a different PAMP that's being detected. So TLR4, LPS, NFkB. That's the key things I want you to remember there. But there are also damage-associated molecular patterns, right? Now, here we have on the left here, we have a parenchymal cell, which is just a regular cell as part of the tissue, right? Now, it undergoes damage. Now, molecules inside the cell then come out of the cell, right? So ATP, and there's another one that's kind of crucial. It's called HMGB1. Now, again, that doesn't mean anything. HMGB1 is not informative, so just learn the acronym there. So HMGB1 and ATP, well then they were inside the cell. In fact, HMGB1 was in the nucleus. That's how deep down in the cell it was. But if a tissue gets attacked and is popped or gets ripped apart through tissue damage, those molecules go from inside the cell to leak out. And now we have damage-associated molecular patterns being leaked out. They are not normally in the extracellular space, so we have evolved receptors designed to detect them, because that tells us that tissue is popping, the cells are being lysed. So ATP and HMGB1 can then bind to receptors on the macrophage or resident immune cell, and then that can lead to changes. Now, this is super interesting. HMGB1 binds to TLR4. That was the guy we just saw in the previous slide that detects LPS. So it's a great evolutionary trick. Evolution loves to repurpose rather than reinvent. It's very difficult to invent something through mutations, but it's very easy to tweak something and repurpose something through mutations. So HMGB1 has obviously undergone a few mutations to make it look more like LPS, so that when it comes out, it can bind to and activate the TLR receptor. So it then goes on to activate NFkB and turn on those cytokines. So we end up gene expression changes and inflammatory signaling molecules. Whereas the P2X7 receptor, it detects that ATP. Now, it actually doesn't cause gene expression changes predominantly. It causes cytosolic changes. So very fast changes in the cytosol. Actually, it opens up a pore, which allows ions to flux everywhere, all over the place. Now, you guys will know potassium's high inside the cell and calcium is high outside the cell. If you create a giant hole, those are going to swap. So potassium's going to leave the cell and calcium's going to come in. And that causes cytosolic changes, which are the secondary signaling molecules. And that can also lead to a quick inflammatory signaling molecule. So why would you have those two pathways? Well, gene expression can be slow, whereas cytosolic changes can be fast. There's also another little problem there. Bacteria and pathogens have learned that we like to turn on genes to try and destroy them. So they often fight back and shut down our protein synthesis, or they target the immune cell to try and kill it. So, and because expression changes are relatively slow, they might kill our immune cell before we can release that signaling molecule. So we need a fast and a slow signaling cascade. So cytosolic changes are often really quick. If you imagine calcium coming in, potassium leaving, that's going to happen in seconds. So we need that fast pathway and that slow pathway in order to ensure that the bacteria don't kill the messenger before it can deliver its message. Don't kill the macrophage before it can go on and tell the body we need to build an immune response right here, which is super interesting, right? So we have those fast and slow responses. So what are these resident innate immune cells? Well, there are macrophages. That's my favorite. They're all throughout your tissue, and sometimes there are specialized macrophages depending on the tissue type. I don't want you to learn these, but microglia are in the brain, and they're a macrophage-like cell, but they're in the brain, so we call them microglia, and they have a unique morphology, similar to Kupffer cells in the liver. But they're all just resident phagocytes that are all part of this sort of umbrella of macrophages. So Kupffer cells and microglia are specialized forms, and they kind of get their own unique little branch off. Now, there are dendritic cells. They're called dendritic because dendrons and dendra comes from the Latin word for branching in trees, and that's because they look spiky and tree-like. If you do any sort of neuroscience, you'll know that the spiky process of a neuron is called a dendrite, and that's a similar thing, right? It's all related to trees. So dendritic cells are spiky, and they're spiky so they can sit still and survey a large area of the tissue with their spikes, okay? So rather than migrating over to see, hey, what's going on over here, they can then just have a branch out there with receptors that are detecting what's going on. And then we have mast cells. Now, these are granulocytes, so they have these densely packed granules that are ready to release an inflammatory signaling molecule called histamine at the drop of a hat, right? So they are primed and ready to release histamine. They're very specialized histamine releases. They also do other stuff, but that's the unique thing about them, and that's why they're so specialized compared to the macrophage. So let's discuss some of the differences between these resident immune cells that are doing the sensing. These guys are doing most of the sensing of the immune system. Location, so they're mostly in tissues throughout the body, whereas dendritic cells are mostly at sites of pathogen exposure, which is epithelial tissues. So they're in your skin, your gut, and your lungs. So macrophages sit all throughout your body, whereas dendritic cells, because they're a bit more specialized to pathogens, are in the epithelial surfaces to hunt out where we are frequently exposed to those pathogens. Mast cells are mostly below epithelial tissues for a very similar reason to the dendritic cells. So they're in your skin, gut, and lungs, but they're mostly just a couple of layers back, but very similar. They are more specialized to pathogen detecting, in particular parasites, rather than bacteria and fungus. They're very specialized to that kind of stuff, and so they need to be where the parasites are. So one of the critical roles you guys would have already covered is antigen presentation, right? So there is the adaptive part of the immune system that does all the memorization and the production of antibodies. And for that process to start, they need to be shown part of the pathogen through antigen presentation. So they gobble up a pathogen, break it up molecularly, and then display it on an MHC2 molecule. So antigen presentation, macrophages do this a lot. Dendritic cells do this super. That's like one of their major roles. And mast cells kind of do this a little bit. It's not really one of their major roles. Dendritic cells are the big antigen displayers. Macrophages also do it too, and they're really good at it. So DAMP and PAMP detection, are these cells coated in those pattern recognition receptors like TLRs that we were talking about? Macrophages, yes, massively. Dendritic cells, yes, massively. Mast cells, yes. Yep, not quite as much. That's not one of their major roles, but it is right up there. They do have pattern recognition receptors, and they do things like release cytokines and stuff like that. Here's one of the major differences. So Fc receptors. So this is the antibody response, and I'll do a little bit more of a deep dive on this. So Fc receptors bind to antibodies, and those antibodies were produced by your adaptive immune system. So the antibodies go out, they bind to the antigen, and then our resident immune cells detect the antibody that has been bound to the antigen. So macrophages, what happens when their Fc receptors are activated? They eat, right? They phagocytose. So if a IgG antibody, for example, has bound to a pathogen and our macrophage Fc receptor detects that antibody, the macrophage will immediately surround and phagocytose that pathogen. Dendritic cells, same thing. It's all about phagocytosis, the Fc receptor activation. This is where mast cells become unique. This is where mast cells become that specialized cell. They undergo degranulation to release histamine, right? So they actually more detect the IgE antibody, which is being released by B cells of your adaptive immune system, and they have bound to a molecule normally secreted by a parasite. And so in response to that, they degranulate and release histamine. So let's do a little bit more of a deep dive on that Fc receptor, because it's so important in distinguishing mast cells from macrophages. Here we have some bacteria. Here we've got IgG antibody. I remember this as it's the good antibody, Ig good. It is the good antibody, and it's a circulating great antibody. It's good. It binds to the pathogen. That's detected on an Fc receptor on a macrophage. That induces phagocytosis, right? And in a mast cell, we have a parasite, and that's bound to IgE. Now, I remember this by allergy. So IgE is also associated with allergies. Histamine, right? Mast cells release histamine. That's allergies. That's why we take antihistamines. So IgE is allergy. It induces allergies, okay? So that binds to the Fc receptor on the mast cell, and that causes degranulation of that histamine, right? But you might say, why do we have a receptor specific for allergies? It's actually more to do with parasites. Now, one of the reasons why this is the case is histamine causes massive mucus production and massive vasodilation, and that's really important for getting rid of parasites. So if we had a parasite in our intestine, if we just pump a bunch of fluid and a bunch of mucus into our intestine, we can flush out that parasitic worm. Okay, so this response is really an adaptive, great response. Right, so that was detection, and so there was a few things there. The pattern recognition receptor is important for detection, but also the Fc receptors are important for pathogen detection. And so TLRs and the Fc receptors are the critical things for sensing PAMPs, pathogen-associated molecular patterns, or antigens in the case of Fc receptors. Up next, we're going to say what happens next, the signaling response. So yeah, we're going to jump in those cytokines and those prostaglandins.